Regulation of RNA processing. It is a process that refers to the mechanisms that control the modification and processing of primary RNA transcripts to generate mature, functional RNA molecules. This process occurs in eukaryotic cells and involves several steps, including 5 capping, splicing, and 3 polyadenylation. First step is capping. The 5 cap is a modified nucleotide structure added to the 5 end of PA mRNAs. It serves multiple functions, including protection against degradation, facilitation of mRNA export from the nucleus, and enhancement of translation efficiency. The addition of the 5 cap is catalyzed by a capping enzyme complex, and its regulation can influence the stability and translation of the mRNA. Second step is alternative polyadenylation. Polyadenylation is the process of adding a poly A tail to the 3 end of PA mRNAs. This modification protects the mRNA from degradation and aids in mRNA export and translation. Alternative polyadenylation refers to the usage of different polyadenylation sites within a gene, resulting in mRNA molecules with different 3 untranslated regions UTRs. This can affect mRNA stability, translation efficiency, and subcellular localization. The choice of polyadenylation site is influenced by cis-acting elements and trans-acting factors that interact with the PI mRNA. Third step is splicing. Introns, non-coding regions within PI mRNAs, are removed through a process called splicing. The exons, coding regions, are joined together to form a mature mRNA molecule that can be translated into a protein. Splicing is carried out by a large RNA protein complex called the spliceosome, which recognizes specific sequences at the exon-intron boundaries. The regulation of splicing involves the binding of splicing regulatory proteins to specific RNA elements, such as splicing enhancers and silencers, located within the PI mRNA. These regulatory proteins can promote or inhibit the recognition of splice sites by the spliceosome, leading to alternative splicing, where different combinations of exons are included or excluded in the final mRNA product. Fourth step is RNA editing. RNA editing is a process in which specific nucleotides within an RNA molecule are altered after transcription. This can result in the conversion of one nucleotide to another, or the insertion or deletion of nucleotides. RNA editing can be catalyzed by enzymes such as adenosine deaminases and can change the coding potential or stability of the RNA. It is a regulatory mechanism that can generate functional diversity from a single gene. Fifth one is RNA stability and decay. The stability of mRNA molecules can be regulated through various mechanisms. Regulatory proteins can bind to specific sequences within the mRNA, known as RNA binding elements, to promote or inhibit mRNA stability. Additionally, non-coding RNAs, such as microRNAs and small interfering RNAs, can recognize complementary sequences within mRNA molecules and trigger their degradation through RNA degradation pathways. And last one is RNA localization. The regulation of RNA processing can also influence the localization of specific mRNA molecules within the cell. Certain RNA processing events, such as alternative splicing and alternative polyadenylation, can affect the recognition of RNA localization signals. These signals guide the mRNA to specific subcellular compartments where they can be translated or undergo localized functions. Thank you for watching, and don't forget to subscribe, like and share.